let us discuss some applications of UV spectroscopy. UV spectroscopy has numerous applications across various fields, including pharmaceutical analysis, environmental monitoring, and material science. UV spectroscopy is a versatile analytical tool used across diverse fields to identify, quantify, and characterize a wide range of substances and materials. For example, in pharmaceuticals, it helps to determine drug purity and concentration, while in environmental science, it monitors UV radiation levels. It is used to study chemical kinetics and analyze the purity of materials. This technique analyzes the interaction of UV and visible light with a compound, allowing us to determine the wavelengths of light absorbed and the resulting color. This technique measures the absorbance of light varying wavelengths through a sample, revealing information about the electronic structure and interaction of molecules. UV spectroscopy finds applications across various scientific fields, including analytical chemistry, biochemistry, environmental science, and pharmaceuticals. It is used for identifying and characterizing molecules, determining their concentration in solutions, and assessing the purity of samples. Additionally, it can be applied for quality control in industries like food and beverage, and for studying chemical kinetics and identifying functional groups. UV visible spectroscopy has several applications, including detection of impurities, quantitative and qualitative analysis of compounds, studying chemical kinetics, identifying functional groups, quantifying pharmaceutical substances, elucidating organic structures, and determining molecular weights. UV spectroscopy, which measures the absorption of UV and visible light by a sample, has diverse applications across fields like analytical chemistry, biochemistry, environmental science, and pharmaceuticals, enabling the identification, quantification, and characterization of various substances. Analytical chemistry. UV spectroscopy is used to identify and quantify molecules, determine impurities, and analyze the purity of samples. Quantitative analysis. Determine the concentration of substances in solutions using Peirce's law, which relates absorptions to concentration, path length, and extinction coefficient. UV absorption spectroscopy can characterize those types of compounds which absorb UV radiation. Identification is done by comparing the absorption spectrum with the spectra of known compounds. UV spectroscopy is used in various fields, including analytical chemistry, to identify and quantify organic compounds, determine their structure, and study their reactions. Qualitative analysis. It can characterize those types of compounds which absorb UV radiation. It can identify compounds by comparing their absorption spectra with known standards. Impurity detection. Identifying and quantifying impurities in samples. Structure elucidation. It helps to identify characteristic absorption bands which can provide information about the structure of organic compounds. Monitoring chemical reactions. By tracking changes in absorptions at a specific wavelength, one can monitor the progress of chemical reactions and determine reaction rates. Determination of acid-base dissociation constants. UV can be used to determine the dissociation constants of acids and bases if the acidic or basic forms absorbs light. quantitative analysis of transition metal ions and conjugated organic compounds. UV spectroscopy is commonly used for the quantitative analysis of these types of compounds. Biochemistry and molecular biology, DNA and RNA analysis. UV spectroscopy can be used to study the properties of DNA, such as its melting temperature and nucleotide composition, and to monitor structural changes in DNA. Assessing the purity of DNA and RNA samples essential for downstream applications like PCR and DNA sequencing. Protein analysis. It can be used to study proteins, including conformational changes and the presence of aromatic amino acids like tryptophan, phenylalanine, and tyrosine. Enzymatic reactions. UV spectroscopy can be used to monitor enzymatic reactions by tracking the formation of products or the consumption of substrates. Environmental science. Water quality monitoring. UV spectroscopy can be used to determine the concentration of pollutants and contaminants, nitrates or phosphates, in water samples. Soil analysis. It can be used to analyze the composition of soil samples, including the presence of fertilizers and other nutrients. Atmospheric chemistry. Studying the composition of the atmosphere and identifying pollutants. Pharmaceuticals and biotechnology. Drug development and quality control. UV spectroscopy is used in the research and quality control stages of drug development to develop active pharmaceutical ingredients, quantify impurities, and perform dissolution testing. Chemical identification and quantification. It can be used to identify and quantify drugs and their metabolites. Drug identification and characterization. Identifying and quantifying drug substances. Pharmaceutical formulation analysis. Ensuring the quality and consistency of pharmaceutical products. Drug metabolism studies. Studying the breakdown of drugs in the body. Food and beverage. Assessing sensory attributes and nutritional components. UV spectroscopy can be used to assess the quality and composition of food and beverages, including non-alcoholic and alcoholic drinks. Beverage analysis. It can be used to identify and quantify specific compounds in beverages such as caffeine and anthocyanins. Purity determination. UV spectroscopy can be used to determine the purity of food products such as olive oil. Detection of impurities. UV absorption spectroscopy is one of the best methods for determination of impurities in organic molecules. Additional peaks can be observed due to impurities in the sample, and it can be compared with that of standard raw material. By also measuring the absorptions at specific wavelength, the impurities can be detected. Spectral elucidation of organic compounds. UV spectroscopy is useful in the spectral elucidation of organic molecules. The presence or absence of unsaturation, the presence of heteroatoms. From the location of peaks and the combination of peaks, it can be concluded that whether the compound is saturated or unsaturated, heteroatoms are present or not, and so on. Biochemistry and molecular biology. DNA and RNA purity and quantification. Assessing the purity of DNA and RNA samples essential for downstream applications like PCR and DNA sequencing. Protein and nucleic acid studies. Analyzing conformational changes in proteins and nucleic acids and studying thermal denaturation, enzymatic reaction monitoring, following the progress of enzymatic reactions by measuring changes in product concentration, detection of functional groups. This technique is used to detect the presence or absence of functional groups in the compound. Absence of a band at particular wavelength regarded as an evidence for absence of particular group. Molecular weight determination. Molecular weights of compounds can be measured spectrophotometrically by preparing the suitable derivatives of these compounds. For example, if we want to determine the molecular weight of amine, then it is converted into amine picrate. Then known concentration of amine picrate is dissolved in a liter of solution and its optical density is measured at lambda max 380 nanometer. Other applications. Forensic science. UV spectroscopy can be used to detect and quantify hemoglobin in skeletal or dried remains. Material characterization. It can be used to determine the transmittance of materials such as sunglasses and fabrics and specific wavelengths. Nanotechnology. UV spectroscopy can be used to characterize the optical properties and the electronic structure of nanoparticles. Food and beverage quality control. Assessing the quality of food and beverages, including checking for impurities and contaminants. Chemical kinetics. One can study the rates and mechanisms of chemical reactions. The kinetics of reaction can also be studied using UV spectroscopy. The UV radiation is passed through the reactant cell and the absorption changes can be observed. Thus, UV spectroscopy is used in various fields, including analytical chemistry, to identify and quantify organic compounds, determine the structure, and study their reactions. Most single bonds, like sigma bonds, such as CC, CH, OH, and CO, have del E values that correspond to light in the deep UV part of the spectrum. They appear colorless to us because light in the visible region, 400 to 700 nanometers, simply does not have enough energy to excite their bonding electrons to an excited state. This is why water is colorless. This is why ethanol is colorless. This is why diethyl ether, hexanes, chloroform, hexanes, chloroform, and a host of other molecules which we encounter both in the lab and in everyday life are colorless, because the del E for the bonding orbital is too large for relatively too low energy photons of visible light to excite them. In fact, UV in the region below 120 nanometer is so energetic it is completely absorbed by atmospheric oxygen and nitrogen. In the UV spectroscopy, organic molecules absorb ultraviolet and visible light, leading to electronic transitions, primarily involving pi and non-bonding electrons, which are excited to higher energy levels. The absorption of ultraviolet light by organic molecules always results in electronic excitation. Bond breaking can subsequently result as an ionization or bond vibration, but none of these processes are guaranteed to result from the absorption of ultraviolet. UV spectroscopy is useful in the structure elucidation of organic compounds. The presence or absence of particular absorption band at a particular wavelength may be regarded as an evidence for the presence or absence of particular chromophore in the compound. Applications. UV spectroscopy used for quantitative analysis, determine the concentration of compounds, structural analysis, identifying functional groups and conjugated systems, monitoring reactions, tracking the progress of chemical reactions, studying protein conformation, observing changes in protein structure.